Hello, Internet. It's a great pleasure to be here to get to tell you about the work we've been doing in smoky Moscow, Idaho, working on bioanalysis of microsamples. Microsampling generally refers to collection of any biological material in small volumes, typically 50 microliters or less. Some microsampling techniques are capable of collecting as little as 10 microliters for quantification. This presentation will focus on bioanalysis of biofluids collected via dried matrix spots and volumetric absorption microsampling devices. Microsampling offers many distinct advantages over conventional sampling for both preclinical and clinical studies. Whole blood microsampling via tail prick in laboratory animals is more ethical than conventional blood draws, reducing the number of animals needed for a study, reducing their discomfort, and allowing increased sampling frequency of the same animal. In human studies, microsampling reduces many barriers to trial enrollment and allows for more inclusive studies. Microsampling via a finger or heel prick is more comfortable for patients who may have an aversion to needles or who cannot be subjected to multiple blood draws, such as infants and the elderly. Additionally, the ease of collection and processing of dried microsamples can greatly expand the number of clinical sites capable of participating in a trial. I will talk in more detail about how the logistics of microsampling reduce barriers to enrollment and compliance in the following slide. To illustrate how microsampling can improve inclusivity of enrollment and improve compliance in human clinical studies, I have outlined the basic workflows of conventional sampling compared to microsampling. For conventional plasma sampling, first a patient must come to the clinic to have blood drawn by a trained phlebotomist. Plasma is then collected from the blood, which requires the clinic to have a centrifuge. At this point, the samples must be kept in an unbroken chain from the time of collection until analysis, the so-called cold chain. The plasma is stored in a freezer at -20 degrees Celsius or preferably at -70 degrees. So the clinic must also have freezers capable of these temperatures dedicated to sample storage. The samples are then shipped in special shipments in biosafety containers on dry ice to the bioanalysis facility, where they again must be stored in expensive freezers until analysis. The need for expensive equipment and the level of training required to enact this sample collection limits the number of clinical sites capable of participating in a trial. Conversely, in a typical microsampling workflow, first, instead of a blood draw, a simple finger or heel prick is used to obtain a drop of blood that is then collected using the microsampling device, which typically contains an absorbent material. This collection requires far less training and specialized personnel than conventional sampling and is even possible to be done at home by the patient in some cases. Next, the sample is typically dried for a few hours at room temperature. No centrifuge and no freezer is required. The drying also inactivates many biohazards, meaning that samples can be packaged and shipped in the normal mail. Many analytes are stable in this form for many months at room temperature, eliminating the need for the cold chain. The factors I just outlined increase the clinics capable of participating in a trial, especially in rural or geographically isolated communities. Closer proximity of clinical sites reduces the travel time and lodging required for participants, and the simplified process reduces time spent at the clinic. Patient logistics and finances are listed in surveys as major barriers to clinical trial enrollment. So, any reduction of these barriers, such as physical distance to the clinic or reduction in missed work, can greatly improve enrollment and compliance. Microsampling is being increasingly accepted as a viable option throughout the drug development process. In the following slides, I will discuss the techniques of dried matrix spotting and volumetric absorption microsampling in more detail, as well as describe work we have done at Alturas Analytics in bioanalysis of small molecules collected by microsampling. One of the oldest and most widely used microsampling techniques is dried blood spotting, which has been used for neonatal screening for decades. In a dried blood spot assay, blood from a heel prick is spotted onto an absorbent card and allowed to dry before storage and analysis. A punching device is then used to obtain a portion of the sample with a known diameter to give a somewhat volumetric amount of sample for extraction and analysis. This technique has all the advantages I spoke of previously in regards to microsampling, but with some caveats. The accuracy of dried blood spot analysis is affected by hematocrit, which is the proportion of blood volume consisting of red blood cells. Differing hematocrit levels contribute to how the blood spreads on the absorbent material, which can affect how volumetric the punch is, leading to over or underestimation of concentration. Dried blood spot assays, therefore, must be validated for hematocrit effects. The same techniques used for dried blood spot analysis can also be applied to any matrix. Plasma can be collected using a DBS card, which eliminates hematocrit effects, but does require centrifugation. At Alturas, we've developed many dried matrix spot methods, which is just an extension of the dried blood spot technique to any other matrix. We have found that clear matrices present a particular challenge in that the matrix is not visible on the card, which makes obtaining an optimal punch where the sample is uniform difficult, leading to poor precision. We've overcome this issue by addition of color indicating dyes to the DBS cards. Here I am showing a card with and without the color indicating dyes. The first circle from the left contains no sample or dye, and the second contains the sample, in this case a synovial fluid. The second two circles both were spotted with color indicating dye prior to sample addition. This final spot contains sample, which can clearly be seen by the blue ring, allowing the analyst to obtain a punch from a location on the card where the sample is uniformly distributed. We achieved a further improvement to this process by adding the internal standard directly to the dye prior to spotting of the sample. In typical DBS analysis, the internal standard is added after the sample is punched during the extraction process. In that scenario, the internal standard can only compensate for the extraction process and sample injection onto the LCMS, or whichever analytical technique is used. Addition of the internal standard from the very beginning allows us to compensate for any variation during the sample preparation and extraction. Adding the internal standard at this stage also allows compensation for sample degradation that may occur during shipping or storage. This should improve overall accuracy and precision of the method. Dried matrix spot extractions are more laborious than conventional plasma and blood analysis because the cards must be prepared and each sample must be individually punched out for analysis. To improve throughput, it is critical to introduce automation whenever possible. We have demonstrated that using conventional liquid handling devices that are already present in bioanalytical labs, that the efficiency of these processes can be improved. Here, I'm showing the addition of the color indicating dye and internal standard being spotted onto the DBS card by a liquid handling robot commonly found in many bioanalytical labs. In addition to using liquid handling robots for dried matrix spot card preparation, we have tried several semi-manual or automated systems for punching the cards. Here, I am showing a semi-manual method for punching the cards directly into pipette tips used by our liquid handling robot, which can then be used directly for extraction as shown on the right. Together, these methods have allowed us to increase the efficiency of our extraction process without sacrificing precision or accuracy, and without purchasing expensive equipment. In volumetric absorption microsampling, or VAMS, a blood spot is collected volumetrically onto the microsampling device. The procedure for collecting these samples is extremely simple and can be done with very little training, much less than dried blood spots. I have pictured here the Mitra microsampling device. These are capable of collecting blood volumetrically, and they are available to collect 10, 20, or 30 microliters. At Alturas, we have used these to measure thyroid hormones at physiologically relevant concentrations. The extraction process is also simpler than the dried blood spot assays, as the tip of the mitra tube can be added directly to the extraction solvent, eliminating the punching step. There is a drawback, though, in that each tip is only useful for a single analysis, so if repeat analysis is expected, then multiple tips must be collected. Blood samples recovered from volumetric absorption microsampling tips displayed less than a 5% volume variation across a range of hematocrits, demonstrating an important improvement over dried blood spot assays. Microsampling offers many benefits, but from a bioanalytical perspective, it does provide some challenges. For microsampling, the analysis must be sensitive, accurate, and high throughput. And this is where many of the challenges come in. Microsamples are by definition micro, which means that we as bioanalysts must be able to achieve biologically relevant limits of quantification with substantially less biological material than we typically have access to. Additionally, the processing of samples from microsampling devices is more laborious than plasma samples, and automation can be difficult. A benefit of microsampling devices is that many analytes are stable at ambient temperatures. This stability should be assessed early in method development. If samples are not stable at ambient temperature, then many of the benefits of microsampling are lost, and conventional sampling should be considered instead. Storage space must also be considered. VAMS devices require a large amount of storage space per sample compared to a regular cryotube. Therefore, a bioanalytical lab must have a large amount of dedicated and controlled ambient storage space, which is usually at a premium. Additional experiments should be done during assay validation to ensure that measurements are not impacted by hematocrit. Although the data I just showed implies that hematocrit experiment may not be necessary for VAMS devices, we still recommend it and have seen some hematocrit effects with VAMS devices in our validations. Another benefit of dried microsamples is the ability to ship in standard mail. However, these samples can be subjected to drastic changes in temperature during shipment, depending on the season. Stability for a duration to cover standard shipment times should be validated for high and low temperatures that exceed the possible shipping temperatures. And if need be, a temperature logger should be used during sample shipment. In our experience, samples should also be shipped and stored with desiccant to control humidity. The use of non-invasive matrices such as tears, sweat, saliva, and milk offer further benefits to patients and to researchers. Non-invasive sampling can be more comfortable for patients, allowing for repeated samplings that would not normally be possible, especially for neonates and the elderly. Non-invasive matrices such as saliva, sweat, tears, and breast milk have proven useful for therapeutic monitoring of many drugs and disease diagnosis, and some have potential for use in PK studies. When considering choosing microsampling of a non-invasive matrix over traditional blood or plasma sampling, several practical matters should be considered. First, the microsampling technique must have sufficient sensitivity to provide biologically relevant data. In addition to small volumes, a drug may be diluted in a non-invasive matrix as compared to plasma. At Alturas, we have often employed microflow LC systems in combination with our mass specs to attain incredibly low limits of quantification, often 10x lower than can be achieved with normal HPLC. This makes microsampling of most analytes possible, and MS technology is constantly improving. Analyte stability is another important consideration. Many of the benefits of microsampling derive from the fact that many analytes are stable at room temperature for long-term storage when collected and dried on a microsampling device. Microsampling devices require more storage space than conventional samples. So if the analyte is not stable and requires refrigeration or freezing, then conventional sampling might be preferable. Further, to generate useful data, concentration of the drug in the non-invasive matrix must correlate to plasma concentrations over a wide range of concentrations. This necessitates that bridging studies be conducted in an early phase of development and means that many drugs are not suitable for analysis from non-invasive matrices. Finally, the more important question to ask is what is actually gained over conventional sampling. The use of non-invasive matrix must provide some real benefit to the subjects and researchers. In the next slides, I will give examples of bioanalysis we have performed using microsampling of tears and saliva. Lacrimal fluid is the go-to matrix for disease diagnosis of ophthalmic disease and monitoring drugs delivered topically by eye drops. Tears demonstrate a high correlation to unbound plasma concentration of a wide range of drugs. Tears are also less prone to contamination by environment, food, or smoking than saliva and sweat. The primary challenge in bioanalysis of tears is collecting sufficient volume. A common theme among all non-invasive matrices I will discuss is that stimulation can increase sample volume, but may also change the chemical composition of the matrix in ways that must be accounted for. The unstimulated rate of tear production is very low, with only 10 microliters present on the eye at any given time. Increased production can be easily stimulated by eye irritation, but this is unpleasant for the subject and causes dilution of the analyte. Tears are typically collected in one of two ways, which are shown on the right. The first is for samples to be collected by holding a capillary tube directly in the conjunctival sac. The sample can then be drawn onto a swab or filter paper. To increase volume collected, a known volume of saline solution can be added to the eye prior to collection, which of course dilutes the sample. A second common method is the Schirmer strip, in which a filter paper is held directly in the conjunctival sac. This method has the advantage of collecting a larger volume of basal tears, but could also cause irritation and stimulate tear production, leading to an underestimation of analyte concentration. Here, we demonstrate the applicability of dried matrix spot analysis to non-invasive matrices by quantifying tobramycin from human tears. Tobramycin is an antibiotic commonly used to treat eye infections. Here we use a color-changing dye to indicate sample location, which can be seen by the purple spot on the card. As you can see, we are able to achieve excellent precision and accuracy using this method with a relevant limit of quantification using only 10 microliters of tears. This method could be used in conjunction with capillary tube collection for real-world sample collection. Saliva is produced continuously by the salivary glands to cover and lubricate oral tissues at a basal flow rate of 0.5 milliliters per minute, but can increase up to 5 milliliters per minute when stimulated to aid in digestion. Drugs are primarily excreted into saliva via passive diffusion in the case of hydrophobic drugs and ultrafiltration in the case of small molecular weight hydrophilic drugs. Hydrophilic drugs larger than 1900 Daltons with low pH are not well excreted into saliva, which limits the usefulness of saliva for quantification of many drug metabolites. Similar to sweat, the pH of saliva is different when stimulated versus unstimulated, meaning that researchers must carefully control their collection processes in order to collect only stimulated or only unstimulated saliva. Stimulation can be achieved by many methods, including by simply putting a small amount of sodium chloride in the mouth. The pH of stimulated saliva more closely matches the pH of plasma, meaning that stimulated saliva can be a good indicator of the unbound fraction of drug in plasma. Saliva can be collected in a number of ways, including passive drooling, spitting, or mouth swabbing. Many commercial collection devices are available, an assortment of which I've shown on the right. With the majority of these devices, saliva is collected by an absorbent swab and then squeezed into the container to provide a larger volume of liquid sample. Other researchers have employed similar dried matrix spotting techniques to which I have demonstrated for tears. Dried matrix spots, of course, have the potential advantage of easier storage and shipment than liquid samples. At Alturas Analytics, we have adapted a volumetric absorption microsampling device, in this case specifically the Mitra microsampling device, for analysis of alternative matrices, including saliva. The Mitra device is typically used for volumetric blood sampling. Samples can be easily collected with high precision at preset volumes. The collection is very simple and can even be done by patients at home. For saliva collection, the Mitra device or something similar could be employed by having the subject spit or drool into a container, and then using the Mitra tip to absorb the sample as shown for blood above. Alternatively, it is possible that a technician could use the Mitra device to collect saliva directly from the mouth. It is important that sample is allowed to be drawn onto the absorptive material in order to collect a quantitative volume. This would require well-constructed instructional materials be developed to aid at-home sampling. Using the Mitra microsampling device, we developed an HPLC MSMS method for quantification of dexamethasone from human saliva. Dexamethasone is a corticosteroid used to treat many inflammatory conditions and has gained recent fame for its utility in treating hospitalized COVID-19 patients. Corticosteroids in general are very amenable to salivary analysis. As relatively small hydrophobic compounds, they are easily excreted into the saliva via passive diffusion. As shown here, salivary dexamethasone concentrations correlate extremely well to plasma concentrations across many orders of magnitude. In the next slide, we'll talk about the extraction procedure. This slide shows our procedure for extracting dexamethasone from saliva using volumetric absorption microsampling. After sample collection, the tip is dried for at least 2 hours, after which it is placed in a 96-well plate alongside standard curve and QC points that were also prepared on the tips. At this point, internal standard and extraction solvent are added, followed by shaking at room temperature for 30 minutes. The extract is then moved to a new plate, evaporated to dryness, and then reconstituted prior to injection on the LCMS. On the right is a sample chromatogram showing a 15 microliter injection of 0.4 nanogram per mil dexamethasone extracted from 20 microliters of saliva on a microtip, we achieved excellent sensitivity comparable to our validated plasma method from only 20 microliters of matrix. The precision and accuracy of the dexamethasone assay is excellent, and importantly, so is the recovery, indicating the analyte is efficiently and completely extracted from the microtip using a relatively simple extraction procedure. Thank you again to the organizers for this great opportunity, and I'd like to take a moment to just acknowledge my colleagues, Jennifer Zimmer, Chad Christensen, and Cody Hawkins. Thank you all for helping me put this together.